Diffusion is the process by which molecules are transported across a cell membrane from a high concentration of molecules to the lower concentration. The cell membrane is made up of a lipid bilayer with the outside part being hydrophobic and the inside being hydrophilic. Nonpolar molecules are able to pass through the lipid bilayer without any help from proteins. Examples being oxygen, nitrogen, carbon dioxide, etc. However, polar molecules are not able to pass through the lipid bilayer without the help of transmembrane proteins. When a protein is used to transport molecules through the membrane, it is called facilitated diffusion. One of the ways for facilitated diffusion is through channel proteins. Channel proteins are transmembrane proteins that go completely through the lipid bilayer with a hydrophobic channel in the middle of them that allows for polar molecules move into the cell. Another transmembrane protein is the ion-gated ion channel protein that is just like the channel proteins, but it has a gate on one side that only opens when a specific molecule bonds to it. This channel is a lot more specific in what molecules it allows to pass through. The last method for facilitated diffusion is with the use of carrier proteins change its own shape when it bonds with a molecule to transport it across the membrane. All of these are passive transports, meaning they move with the concentration gradient from high to low and do not require energy to do so. Osmosis is the movement of water across a membrane through a protein called aquaporin from a high concentration to a low concentration until equilibrium is reached. Aquaporin is a type of channel protein. Free water is water that is not dissolving or bonded to any other molecule. Free water is the only molecule that moves across the membrane in osmosis. When free water is equal within the cell and on the outside of it, that is called an isotonic solution. When there is more free water within the cell than on the outside, the water will move from the inside to the outside, thus shriveling the cell. This type of solution is called a hypertonic solution. When there is less free water inside the cell and more on the outside, water will move into the cell from the outside, thus swelling the cell and eventually causing it to lyse or burst. This type of solution is called a hypotonic solution. Animal cells function best in an isotonic solution. If it is one of the other two, they are likely to die. Plant cells most prefer the hypotonic solution because it increases their turgor pressure, or the pressure the cell wall puts on the incoming water. If the plant cell has an isotonic solution, the plant cell will become flaccid and will lose turgor pressure. If the plant cell has a hypertonic solution, the plant cell will die due to plasmolysis, which is when the cell membrane detaches from the cell wall. All of these are based on tonicity, which is the ability to gain or lose water. Another type of cross-membrane transportation is active transport, which is when molecules move through the membrane from a low concentration to a high concentration using a protein pump. This type of transport requires energy. One example is a sodium-potassium pump, which pumps sodium out of the cell and potassium into the cell's cytoplasm. These are pumped against the concentration gradient. Another type of transportation is called bulk transport, which requires energy and has two forms. One form is exocytosis, which is when the cell secretes waste by bonding to the cell's membrane and getting flushed out into the surrounding area. Endocytosis is when the cell engulfs large molecules or nutrients. There are three types of endocytosis, the first being phagocytosis. Phagocytosis is when the cell membrane changes its shape to engulf a food particle by surrounding it. Then lysosomes fuse to it and break it down, changing it to a food vacuole. The second type is called pinocytosis, which is when the cell membrane engulfs extracellular fluid to get the molecules that are dissolved in the fluid. The third type is called receptor-mediated endocytosis, which is when the membrane engulfs molecules using receptor proteins which will only bond to a specific molecule. The molecule it wants to take is classified as a ligand.